In the translational group, we're of course interested in really identifying those patients where our therapy is going to work, but also to identify mechanisms of treatment resistance and also mechanisms of toxicity. So we're doing a lot of work to understand who is most at risk for grade 3 neurologic toxicity or higher. That we're doing extensive correlative analysis using over 1,400 covariates taken from biomarker sample analysis or from the clinical trial data that's coming back from the sites to try to figure out an algorithm or a way to predict who is more likely to develop neurologic toxicity. This is important because if we can identify patients that are at higher risk, we can keep them closer to the hospital. On the other hand, if we can find patients that are less likely to have a severe adverse event, they're more likely to go home. And one of our goals is to try to move this to an outpatient therapy. We're not there yet, but we're doing a considerable amount of work in that area. We're also trying to understand why some patients either fail to respond, so they're primary refractory to CAR T, or they respond and then at some point in time later progress. And so data that we presented at SOHO yesterday in the poster session was highlighting features of the tumor microenvironment in the immune landscape that create what we call a hot signature or a favorable immune microenvironment, or a cold signature, which provides a barrier to CAR T cell entry. And what we're finding is that there are a subset of patients in our ZUMA-1 study that had a cold signature. 4 out of those 5 patients did not respond as best response to axi-cel. 1 patient did. It's a small data set. We're building on this across our ZUMA studies to try to understand the microenvironment features. We're also taking a hard look at the product and the constituents of the product to understand which features of the CAR T cell product may be deleterious and lead to an adverse event, or may be favorable in our promoting response to the therapy. So we're looking at all possible angles: tumor, product, patient characteristics, pharmacokinetic profile, and pharmacodynamic profile, to really try to integrate all of these findings to develop hopefully a predictive algorithm to understand who is more likely to respond or who's more likely to become sick when they're treated with the therapy. The spin-off is that a lot of this information also gets fed back to our Kite research group to inform them on how to design the next generation of CAR T cells. And so Translational Sciences group at Kite is contributing towards generation of data that will go back to our research group to tell them this is what a good product should look like, this is what a poor product looks like, and we can then find ways to better engineer our CAR T cells for those next generation products. So we're really excited about using data coming off of our clinical trials to not only address where we are now in the commercial setting, but what's the future going to look like. And so this is something that we'll continue to invest quite a bit in and work very hard to achieve.